Everybody, myself Halawat Ramesh. Welcome back to my YouTube channel. After long time, we will back to our YouTube channel. So especially today we will talk about ICP mass spectroscopy. In the sense, inductively coupled plasma mass spectroscopy. The basic information we will discuss, and the next class we will go through the detail about the ICP mass spectroscopy. Now we will go through the slide by slides what is ICP mass spectroscopy and some basics and fundamental of spectroscopy and chromatography techniques, and also we will discuss some hyphenated techniques. Now you can see the first one slide. Before we are going to discuss about the ICP mass spectroscopy, now we need to know the hyphenated technique. So what is hyphenated techniques? See, the couple of decades ago, the Hirschfeld introduced the term of hyphenation, the referred to on the line combination of a separation techniques and one or more spectroscopic detection techniques. So hyphenated techniques refer to system where two or more analytical techniques are combined together in the tandem arrange of in order to increase the analytical specificity. So the main reason the combining techniques together is increasing the sensitivity of detection and the selectivity of separation and improve the reliability of identification. Then we can go for the what are the some hyphenated techniques. You can see here UV spectroscopy combined with DSC and DSC combined with FTIR, LCMS, MOS, GCMS, Raman spectroscopy, TGA, STA and DMA and HGGC. These are the few of the example of hyphenated techniques. Now you can see here before we are going to discuss about that ICP mass spectroscopy and hyphenated techniques, most of the based on the chromatography. So what is chromatography? In all that, chromatography is one of the separation techniques. Simply you can say in separation techniques, in chromatography there is a two phases. One is the mobile phase as another one is the stationary phase. So the mobile phase we will inject the samples and stationary phase we will refer to as that mixture of the through mobile phase to carry into the sample mixture through the reference of a stationary phase. So in liquid gas chromatography LC, in mobile phase has used as a solvent, and gas chromatography is mobile phase inert gas such as the helium. So the stationary phase is usually chemical that can selectively attract the component in a sample mixture. So the stationary phase is usually contained in a tube of some sort. This tube is referred to as a column. So column can be gas or stainless steel of various dimension. Now you can see here the mixture of components in mobile phase interact with the stationary phase. Each compound in the mixture interact with the different rates. Now you can see that changing characteristics of the mobile phase and the stationary phase, different mixture of chemical can be separated from through the chromatography technique. Now you can see the hyphenated techniques combine with the chromatographic with the spectral methods to exploit the advantage of two things. One is the chromatography is produce pure and nearly pure fraction of chemical components in a mixture. Now you can see the spectroscopy. Spectroscopy produce the selective information for identification of using standard or library spectrum. Now you can see here especially the chromatograph and interface, ion source and the mass analyzer, ion detection, then data analysis. These are the few of the basics chromatographic mass analysis components like pathway. Now we can go for the mass spectroscopic techniques is used to support the hyphenated techniques. How it will be mass spectroscopy effect on that hyphenated techniques? You can see the supporting. You can see the ionization and mass analyzer. You can see ESI, electron spray interface, time of differ, and that APCI, atmospheric pressure chemical ionization, and ion trap mass spectrometer, and MALDI and quadrupole. Here is chemical analyzer, here ion trap, and electron ionization, here Fourier transform. These are the ionization and mass analyzer to supportive of the hyphenated techniques. Now you can see here what is the electrospray, ESI. Electrospray means the high mass capability or high mass accuracy and capable to analyze the most of the ions, both organic as well as the inorganic compounds. You can see here Few of that the liquid flow rate in 0.1 milli mm, id the hypodynamic the needle which is held to high voltage like 6 kV. So the liquid spilled from the needle tip where the Coulombic repulsion cause its spread out has a pump of charged droplets. So the solvent evaporated rapidly from small droplets. You can see here this is the molecules and evaporation goes to this like, and then after a breakup you can see that bigger molecules to smaller molecules like ions. That is called breakup. Then you can see here, you can see this picture, you can clearly understood that molecules are atoms and spraying into the electron small molecules. So this is that electrospray ESI you will understood from this picture. Now you can see here the rough diagram of this sample input from the HPLC, then after first quadrupole analyzer, then collision cell, then after the second quadrupole analyzer. Then you can see here the inductively coupled plasma mass spectroscopy, ICP. What is the actual the method of ICP? What is the main concept of ICP mass spectroscopy? So inductively coupled plasma mass spectroscopy is a type of mass spectroscopy which capable of detecting metals and non-metals of concentration has parts for billions. That point is very important, especially in ICP mass spectroscopy. So mass spectrometer is analytical techniques to analyze the chemical species and sorts the ions based on their mass to charge ratio. This point is very important. And then you can see here when the analysis needs to be identified which element something is made up of, they will often use of techniques called as inductively coupled mass spectroscopy. So ICP mass is elemental analysis technique. Simply you can say ICP is one of the elemental analysis analytical techniques. Then you can see here how working principle. Now you can see here the working principle of mass spectroscopy, ICP mass spectroscopy, how it works. So inductively coupled mass spectroscopy, it is the analytical instrument used to elemental determination. The sampled materials introduced into high energy argon plasma that consist of electrons and positively charged argon ions. In plasma, the material split into indivisible atoms. Especially, what is the main role of plasma? The material split into indivisible atoms. That is the working principle of ICP mass spectroscopy. Then you can say, how does ICP-MS work? An ICP-MS instrument uses the plasma ICP to analyze the elements in a sample and then measure the ions using mass spectrometer. The main component of the single quadrupole ICP-MS instrument are sample introduction system to form a fine aerosol moist from the liquid sample. That is the actual working principle of the ICP mass spectroscopy. Then you can see here the plasma is conducting gaseous mixture conducting the significant concentration of cations and electrons. So inductively coupled plasma source, you can see here this picture, you will understood how what is the argon ions once you will from this picture, and you can see the plasma create a something radio frequency, right? And then after the molecule will be mobilized, then ions, then you will be go for the mass to charge ions. Then you can see here next slide, nobilizer of ICP. So solution samples are introduced in the plasma through the nobilizer. So nobilizer is the main role is through the opening of the surrounding of the capillary concentrating. These cause to reduce the pressure at the tip of aspiration of the sample. So the nobilizer will to control reduce the pressure of the tip of this the samples. That is the main role of the nobilizer in ICP. Then you can see here the ICP touch. That's we was collected this image from our instrument. Our lab is having the ICP mass spectroscopy. Then you can see ICP torchlight. Then you can see here the block diagram of ICP mass spectroscopy. So first one is the detector, quadrupole and ion lens and interface, torch, plasma, ICP coil. Then you can see the plasma gas, auxiliary gas and carrier gas and sample and spray chamber, nobilizer, ion sense and detector, quadrupole, interface and plasma. Then you can see this is the picture already we seen in the second quadrupole analyzer collision of the cell. Then you can see this is the simple diagram of ICP mass spectroscopy, and you can see working of ICP mass spectroscopy. These few points are very important. You can see that schematic diagram of ICP mass spectroscopy, the meter system. You can see the sample introduced in ICP with the nebulizer. The ICP heat the sample approximately in 7000 Kelvin, and the solids are evaporated, are atomized and ionized. So the fraction of sample entered into the optical through the differentially pumped region at the approximate in the 1 tera. The ion optics incorporated in a central stop to reduce the photon noise. So the ions then enter into the quadrupole through the short radio frequency. The exit is the channel of electron multiplier. So the detector is mounted off axis to further reduce the photon noise from the plasma. That point is very important. So this is that one of the schematic diagram of ICP mass spectrum system. Then you can see the few of the samples we analyze. This is the best example of spectra, the picked in the leaf digest and arched in the leaf in copper, and you can see the different spectra. We can see the mass versus and the counting of the plasma. Then you can see this is the picture. This can identify more than 75 elements through using the elemental by the atomic spectroscopy and ICP mass spectroscopy. These are the table to represent the elements that can be analyzed by the ICP, the detection limit or the regularly in order to PPT. So parts per trillion, and the equivalent of nanogram for diameters in cube. Then you can see this is the simple diagram of the ICP mass spectroscopy. This is our lab setup of ICP mass spectroscopy, and this is the actual that internal parts. You can see here the ICP mass spectroscopy how looks like the 4th generation reaction system, and this is that vacuum system how is the generating. This is that internal parts of the ICP mass spectroscopy. Then you can see the simplified schematic. Then the functions component, the sample introduced, and then after plasma ion source, then after ion lens, then after collision reaction cell, then is the quadrupole in the mass spectroscopy, then after the detector. This is the one of the schematic functional components. You can see here this is the first steps to last step. You easily will understood what is the main principle of ICP mass spectroscopy. Then you can see here, actually this is the diagram, the sample will preparation, then sample will introduce, the ion source in ICP, then after focus and separate, and then after vacuum system in mass filter, then mass spectroscopy, then detector and data analysis. Actually the main role is the main component is sample introduced to, then after moves into the ion source, then after the mass analyzer, then after detection unit, data analysis. This is one of the main components of the ICP mass spectroscopy. Then you can see few of that full name of the ICP mass analysis, that Thermo Fisher scientific company that throughout India worldwide they are making the different hyphenated techniques, and this is the one model ICP-MS, ICA, PRQ, the quantitative and quantitative PPB in the elemental analysis use. This is the one of the instrument. Our lab is having these facilities, ICP mass spectroscopy. So, and this is the ICAR and MRI National Meat Research Center. They recently purchase of this ICP mass spectroscopy. Then you can see here these are few of the application, how is used for, what is the purpose, food industry, pharmaceutical industry, organic compound and organic metallic compound, gasoline fuels and coal and cake. These the different purpose they are using for the detection of element. Then you can see this is that our user instructions and the user charge also. You can say liquid samples which does not require any preparation. You can see that 2000 rupees for the sample, and for 23 elements we will be analyzed, and 300 for the extra elements. Okay, you can see the sample required for the preparation of using microwave digestion. So this is the 3500 per sample for 23 elements detection, and we will give this is the including the 18% of GST. So this you can see here, this is that our lab vacuum system, and this is the another one system of Elegant, and you can have this is that Thermo Fisher Scientific Germany also they introduced this ICP mass spectroscopy. Then working principle and the user manual, and you can see the features, and then this is the ICP mass calibration graph. Then you can see the few of the points about the how you will calibration, and these are the for future Allegiant ICP mass 790 with the Umini. Then after is the sample preparation, the qualitative and quantitative. Solid is liquid 100 to 500 mg, and liquid is the 5 to 10 ml is enough for 2 analysis. and these are the film cadmium, chromium, and this is a elemental analysis sample, and those are the disease, how it they affect on this, and this is the spectrum, and like, share, comment, and like